Now let's look at the RNAs. RNA has a very similar structure to DNA. DNA case, we had the ATCG, and the carbohydrate was 5-membered the sugar, but it doesn't have a 2' the hydroxyl group. In comparison, RNA has this hydroxyl group at the 2 positions. That's why the sugar difference here, RNA is based on ribose, and then DNA is based on deoxyribose. Seems like it's other than that, it's almost similar, almost the same to each other. And base pair is also, they share the G, C, and A. Only T has slightly difference. DNA has a T, and RNA has a U, uracil. Only difference here is one methyl group is not existing in U. So that means DNA can make its partnering with another DNA strand. How about DNA to RNA? What do you think? They can bind or not? Yes, they do. So the binding side is this side of a hydrogen bonding forming side, G to C, and then C to G, and A here, this side, and bind to T side here, or U side, and then U here, the T here, they can bind to A through this side. That's why DNA can bind to RNA, and RNA bind to another RNA too if the sequence is complementary to each other. Interestingly, RNA binding affinity to the other RNA is actually stronger than DNA to DNA. Then how about the DNA to RNA, the hybrid of the sequence? Actually, that's in the middle. Interesting, isn't it? Now the structural difference only for this 3 position of OH seems like this is a very small difference, but actually extra existence of this hydroxyl group make the stability of RNA is very different from DNA. DNA molecule, as we know, is very stable. So sometimes the DNA molecule can survive for over billions of years. So that's why from fossil, you can extract, in principle, extract out the DNA and then amplify that by PCR, then maybe utilize it to make maybe a dinosaur, right? So that's kind of the scientific background of Jurassic Park, the movie. Now RNA case, because of this extra hydroxyl group, this RNA is much more unstable. Why is it? Can you guess why? These hydroxyl groups can attack this phosphate. Then intramolecular hydrolysis is also possible. Actually, in this case, should be the alcoholysis. Anyway, this cleavage the reaction is the very possible, and also these extra the hydroxyl groups can attract other cofactors like the metal. Then it can catalyze the cleavage reaction too. That's why DNA is more than 1,000,000 times stable than RNA structures. That's why we utilize the DNA as our genomic structure, RNA as a temporary information, for example, mRNA, messenger RNA. So this is unstable, then easier to degrade, and then easier to remove. Transcription. From DNA strand and then making the RNA, especially in this case mRNA, this procedure we call transcription. Now the synthesis is a similar way to DNA replication, starting from the 5' and then utilizing this RNA polymerase, it is synthesizing to this strand. Then genetic information here is this RNA sequence should be genetic information. Then how about this strand? In this case, this RNA sequence should be same with this another side of this sequence. So that's why this sequence, this strand we call this is a coding strand. Then how about this strand? This strand is a template strand, but that's a complementary sequence. That's why, in a sense, if you say this is a sense sequence, this strand is anti-sense strand. Then how about this mRNA? Is it sense or anti-sense? It should be sense, right? So sense sequence, anti-sense sequence means this is a complementary sequence to the other strand. So very interestingly, if we define the gene of course, this coding strand, this is a gene part. The sequence of this part is a gene, and that is the same with this mRNA. But we utilize its complementary sequence as the template to make genetic information of the mRNA. It's a very interesting system. Now for the transcription, we need this enzyme. RNase are the polymerase. So RNA polymerase, now this orange line is a DNA. Now to make RNA, you need to choose one strand. One strand should be template the strand. First step should be open up the DNA into two strands, and then synthesis is happening from here moving to this strand. So this is the X-ray structure of one of those famous T7 RNA polymerase. Now this strand versus this strand, which strand has sense the information? That's this strand, right? So this strand should be antisense or template the strand. Now let's look at dynamic of the movie for RNA synthesis. The synthesis of a new RNA strand requires several different other factors. First of all, this is the DNA, and then they need the activating other proteins, so-called the transcription factors. They recognize those initiating part of that RNA synthesis and bind and to initiate the process. Then other proteins, the most important thing should be this is RNA polymerase bind here, but another helper proteins should come together and make the complex. Not only this area, remote area, there is another activating unit here. So this kind of enhancer or activators should help this regulatory part, the upstream of this DNA sequences, and then they should help and also initiate this RNA synthesis. If everything is ready, now the RNA synthesis can start. So released RNA polymerase now move around. It's like sliding on DNA and start the RNA synthesis. Sliding DNA, this RNA polymerase collect these building blocks of RNA, the ATP, and then synthesize those new RNA to these directions. Now, this is originally double-stranded DNA, and then first step should be open up this into the single strand, and then utilize only one of them. That strand should be template strand, right? And then utilizing that, and then synthesize this RNA, and then release the product to the outside. Now this is a very highly dynamic process, collecting the building blocks and synthesize those new RNAs. So until it reaches to the end point, it continuously synthesizes those RNA. Once it reaches to the stop codon site, now that synthesis is stopped, and then synthesized RNA is released. This released RNA should be matured and then ready to go out of the nucleus. Here is another electron microscopy the picture for transcriptions. Now at the center here, this is the DNAs. Now if you imagine, let's say the synthesis of RNA is happening at the same time in multi-site. Now which site is the starting point and which site is the end point, do you think? This one or this one? Yeah, start site should be as a begin site. It should have a shorter chains, and then multi-RNA polymerase is bound here. It's not only one sliding like this. Actually, the hundreds of them is bound here, moving together. And this is almost end position, so that's why the product is getting longer and longer. Actually, this picture is showing that the synthesis of ribosomal RNA. So once those RNAs are formed, and then at the end they start to fold into structured forms. Once mRNA is synthesized, they need to be matured. The first product, directly transcribed from DNA, it has exons, but at the same time it has intron structures intervening these exon sequences. That's why this intron should be removed by splicing process and eventually make mature mRNA, which has only exon sequences. Once this mRNA is matured and then secreted into cytosol, then they are ready to go for protein synthesis. Now let's study about the concept of a cDNA library. Mature mRNA has an exon sequence here, and then 3' UTL area, they have poly A structures. And utilizing this poly A, now it's possible to design primer to make the complementary the strand. If it's a poly A here, then what can be the primary structure? Yes, it should be poly T. So utilizing this poly T, now utilizing reverse transcriptase, using RNA as a template and synthesizing DNA. Our body does not have this enzyme, but there is some RNA-based the virus has this enzyme. Utilizing those enzymes, we can synthesize this DNA sequence, which is a complementary to this mRNA. It's a complementary sequence to the codon, or complementary sequence to the genetic information. That's why this DNA set we call cDNA, or complementary the DNA. After synthesizing this new strand, either dissociate or utilizing RNase H, then RNase H can cleave off this RNA structure selectively. Then we can get this set of new DNA the set, which has the complementary sequence of mRNAs. If you collect this whole collection of complementary the DNA from one set of mRNA, this collection we call cDNA library. By PCR amplification, we can make the higher amount of this library, and if you put them again into the in vitro transcription, utilizing this DNA as a template, now making mRNA, then we have a chance to make many copies of mRNA. Eventually, we can make, using this mRNA, we can make proteins. Now, this set of cDNA we can call transcriptome, because this is transcribed and eventually which make the proteins. Then how is it different from exome? We have studied the exome before. It seems like quite similar to each other, but it's not exactly the same. What can be the difference? So here is a quiz 1. Please study the difference between transcriptome and exome and put up your answer on IVLE using the poll function. If we are in the battle and our enemy has these tanks, one way to destroy the enemy is maybe destroy these tanks, the machines. But if you think the information to make these tanks is from their headquarters, maybe smarter way is, rather than just individually break up those tanks, maybe attack this information center. Then they cannot send out that information to make those tanks. How about it? So if you compare this with the cell situation, DNA make RNA, and then RNA make eventually protein. So that's why if you want to attack these protein cells, one approach can be, rather than directly attacking this protein, maybe we can attack this DNA or RNA. Then how can you attack those DNA or RNA? We can utilize the nature's the property. If you have exactly complementary sequence to its sense sequence, then they can bind to each other, and then they can interfere their original functions. This idea we call antisense. And then to make it easy to send them into the cells, maybe we can make it into the short nucleotide chain. That's why this kind of drug candidate molecules we call antisense oligonucleotide. So the idea is, if you add those kind of antisense oligonucleotide into the cell, they can bind to its complementary sequence. Let's say if the target is mRNA, they bind to the mRNA, and then they block their translation to make the protein. Then you can block those protein synthesis. So this is the idea of antisense. And this antisense the compound or the concept has been used to develop new the drug for some time. During the antisense experiment, scientists found very interesting new phenomena, so-called siRNA. In the experiment, they used C. elegans as a model, and this is a C. elegans embryo. If you don't express the protein, then this C. elegans does not have any protein signal here. Once you transfect that embryo, then the embryo starts to generate the proteins. This black color is a protein synthesis. Now here, they tried to put in antisense oligonucleotide into this embryo to suppress this protein synthesis. And in one experiment, yes, the antisense showed the relatively good effect, which is suppressing the synthesis of this protein here. As a control experiment, they added not antisense, they added a sense sequence. Sense sequence means it has the same sequence with the mRNA, but the length is the same as of this the antisense oligonucleotide. That's why in that control experiment, they cannot bind to mRNA. That's why they expected to see this kind of full expression of the protein. But the result, actually they found that sense strand, which should have a negative effect, actually that worked much better than antisense itself. And they tried to figure out this strange observation, and later they found that actually they made a mistake, and they thought they put only sense sequence here. Actually, they pipetting together antisense and sense together, and then they make a double-strand oligonucleotide, and then those double-strand oligonucleotide has much stronger activity than antisense itself. This was the discovery story of this siRNA, small interfering other RNAs. That means if you add this small oligonucleotide, if it is a double-strand structure rather than antisense, it has much stronger inhibiting activity for protein synthesis. Once double-strand RNA is introduced into the cell, the RNA is processed by the protein dicer. Dicer cleave off this other RNA into 20 to 22 size of oligonucleotide size, and then this dicer transfer that processed RNA into another protein so-called argonaute. Argonaute dissociated this two-strand RNA, and then one of them it keeps as a guide RNA, the other one, released one, it is degraded by other enzymes. Now, by complexing with the proteins, this argonaute make RISC. It's an RNA-induced silencing complex, and then RISC contains this one guide RNA, and then find it's exactly matching the sequence from mRNA. Once it finds its mRNA, then it is bind to this mRNA. Eventually, this RISC cleave off this mRNA, eventually killing the signal of protein synthesis. Once mRNA is cleaved here, it can be digested by other enzymes, and then the signal is off. This kind of a double helical structure of RNA was not common in our body, so we thought those are maybe introduced by RNA virus, and our body developed the defense mechanism utilizing this dicer and RISC. So that was the origin of this siRNA, we thought. But later, utilizing this concept, scientists can synthesize those double helical other RNA and utilize it for the biological other studies. Let's summarize those RNA structures. The first RNA, we said, this is a coding RNA, which make eventually protein, so this is a gene. So this RNA we call messenger RNA or mRNA. And there was different type of non-coding RNAs. Here the coding means making protein. Even though they don't make protein, some of them is highly functional. The examples were ribosomal RNA. Ribosomal RNA is catalytic subunit of protein synthesis. And the other RNA is a tRNA, which carries amino acid for the DNA, the protein synthesis we are going to study later. Today we studied the siRNA, small interfering RNA. siRNA was silencing those messenger RNA signals. siRNA is one kind of RNAi. RNAi is RNA interference. In the beginning we thought siRNA is quite a unique phenomena, but later, very similar mechanism is universally ongoing in our body too. So those RNAs are microRNAs. Similar size to siRNA, but they have some differences. So this is second quiz I give it to you. Go to the references, Wikipedia or YouTube. Study the difference between microRNA and siRNA and then put up your answer into IBLE. Many more different types of RNA is under discovery. piRNA or ribosomal switch, which is a conformational, the regulating of the RNAs. And introns. We didn't say much about the intron, but some of those intron has a catalytic activity. That's why it is called as a ribozyme. So RNA and enzyme is a complex word. Many different kind of the new RNA is coming up. And there is one hypothesis. Maybe before the DNA and protein has a dual function in our body, maybe RNA was acting as a genetic information, and at the same time, RNA molecule is a catalytic subunit together. So, information and machinery was stored in one unit, one kind of molecules, only in RNA. Currently, we don't have that kind of evidence, but this is very attractive hypothesis. Before DNA protein or world, there was maybe the RNA world, the life form of ancient the Earth.